Lesson 3b, DNA replication. Recall that when cells divide through a process called mitosis, each of the two cells produced, which we call the daughter cells, will have the same number of chromosomes as the initial cell, which we call the mother cell. In order for this to happen, the DNA in the mother cell must be duplicated before it divides, or the subsequent daughter cells will only have half the amount of DNA as the mother cell. This process of copying the DNA for cell division is called DNA replication, and it ensures that the two daughter cells that form from mitosis are genetically identical to the initial mother cell. Replication occurs before mitosis, during the late part of interphase. The process requires a protein enzyme called helicase to come along and unwind and unzip the DNA molecule into two DNA strands called a replication fork. Another enzyme called DNA polymerase III then brings in complementary DNA nucleotides and polymerizes them into a long chain along both strands of DNA through a chemical process called dehydration synthesis. The issue is that DNA can only be added in one direction, along the pre-existing DNA strands. The DNA polymerase III reads the mother strand in the 3 to 5' direction. Because the two strands that comprise a DNA molecule are complementary, they actually run opposite to one another in orientation, in which the sugar and phosphates are polymerized together. This makes one strand literally upside down relative to the other strand. DNA polymerase III can only read strands from the 3 to 5' end. So the strand that continuously exposes the 5' end can grow continuously. We call this the leading strand. The second strand is called the lagging strand and will continue to expose its 3' end. So it must be copied in short sections called Okazaki fragments. This requires the help of a special enzyme called RNA primase that will place a few RNA nucleotides to allow for DNA polymerase III to attach and to begin placing complementary nucleotides while reading the parent DNA strand from the 3 to 5' direction. The new Okazaki fragment of DNA will grow in the 5 to 3' direction. Once the Okazaki fragment created runs into the end of the previous Okazaki fragment, DNA polymerase III falls off, and DNA polymerase I comes along to delete the RNA primer sequence. Once the RNA sequence has been removed, another enzyme called DNA ligase comes along to polymerize the two short sequences into a longer chain of DNA. Now that the cell has created a copy of DNA, identified by the X-shaped chromosomes, the cell will proceed through the mitotic steps of cell division called prophase, metaphase, anaphase, and telophase, where two genetically identical daughter cells, each containing one strand of DNA from the mother cell, and the other strand being brand new. Watch the following videos a few times to clarify this process of DNA replication.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.